From surviving rocket launches in the 60s to having their brains mapped in stunning detail, mice remain at the forefront of science. A new study unveils the intricate wiring of a mouse brain, offering clues to our own. More details in this story.
Mice before men, that's the big idea, and they selected Sally, Amy and Mo to make the trip. Amy's saddled with an instrument to record her reactions during the spaceflight. They were placed in a miniature version of the sort of capsule that would be used for a man. The nose cone of an Atlas missile was to be the carrier.
Mice have always been at the forefront of human scientific research.
The time had come for the blast-off. This is it, said the mice.
Back in the 1960s, 3 mice were fired over 5,000 miles in a missile as part of a rocket testing. But have you ever wondered what exactly was going on in their tiny brains as they were blasted off? Well, a new study might have some answers. Scientists in the US have produced intricate diagrams of the wiring inside a mouse brain.
The brain is kind of structurally like a giant ball of spaghetti, right? You have all of these wires, they're all completely intertangled. It's a complete mess, except for at the ends of those pieces of spaghetti, that's where they actually talk to each other. And sure, we knew that the brain is structured like that, but there are actually many different kinds of spaghetti. There are all of these different colors and all these different flavors, and we don't actually know what are all the colors and the flavors, and we don't know which of them are talking to each other.
The massive dataset marks a major step towards unraveling the mystery of how human brains work. Researchers are hoping this new map will shed light on how these vast circuits operate and how their malfunction might contribute to conditions like Alzheimer's and autism.
You look at these kinds of pictures of the brain and you get a sense of how complicated you are. We're looking at, one tiny part, 1/500th of a mouse's brain. And the beauty and complexity that you can see in these actual neurons and the hundreds of millions of connections between them.
This significant leap in brain mapping was achieved through a collaborative effort involving multiple research institutions. The initial stage involved scientists showing a genetically modified mouse various video clips, including excerpts from the popular Hollywood film, The Matrix. As the mice enjoyed the action movie, scientists then monitored its visual cortex activity using a laser microscope. The crucial final step involved aligning this detailed map with the original functional data from the mouse's brain, creating a powerful tool for understanding brain activity.
And so they systematically looked at how these cells flash or become active when the mouse, in this case that we were doing the experiment on, was looking at a bunch of movies. And so we have a whole host of data before we cut up the brain into tiny sections about how these neurons respond to visual stimuli. And then we took the brain of that mouse, and that's the one that we sectioned up and then measured how these cells are connected to. So we have in this data set both the structure of the brain and measurements about how those exact individual cells were responding to visual stimuli.
Calling this a foundational advance, much like the initial Human Genome Project, the researchers are now setting their sights on an even larger prize: mapping the entire mouse brain.